Microbiology is the study of an invisible world around us. We're surrounded by a host of tiny microscopic organisms that we can't see. Yet these organisms can cause complete havoc. They can cause devastating pandemics. They can also be put to very good use. They're used in recombinant DNA. They're little mini factories used in beer manufacture, bread making, antibiotic production. However, you need to think how we're going to work with these microorganisms and how we're going to work safely with these microorganisms. In this series of videos, we've put together some clips of the key procedures used in microbiology. Practical microbiology is critical to this science. When working in the microbiology lab, there's 3 reasons why you have to use good microbial practice. The first of these, of course, is safety. You need to prevent yourself from becoming contaminated from the microbe you're handling. You also need to consider those around you, so you have to prevent dissemination of the microbe into the environment. You don't want the person next to you to come down with the disease from the microbe you're working with. There's one other reason that may not be immediately evident, and that is you need to protect the sample that you're working with. If you've just been on an expedition to Mars or to the polar cap to Antarctica and you've come back with a sample of microbes that may grow in an extreme environment, the last thing you want to do is to isolate Staphylococcus aureus that grows in your hand and say this is the microbe you've isolated from this extreme environment. So containment of the microbe in the tube you're working with or the plate you're working with, as well as protection of your sample from microbes on your body. Okay, the first thing you have to do when working safely in the microbiology lab is to wear a lab coat. In microbiology labs, the lab coats are special coats. They have elastic around the arms so that nothing goes up your arms, and they tie right up to your neck, and you should tie the front of the lab coat so that it's completely tied. Now, there's certain rules we have working in the microbiology lab. In addition to wearing your lab coat, you never ever bring any food or drink into the lab. Nothing should touch your mouth. No chewing gum. You don't put your fingers in the mouth. You don't put the end of your pen in your mouth. That completely avoids risk. There should, of course, be no contamination on your hands. If you should at any point spill anything on your hands, you would then come to the basin, wash your hands, disinfecting them before you continue working again. If you're thirsty during the practical, what you do is you come here, you take off your lab coat, you disinfect your hands, you go out and take a drink of water outside the lab and then come back in again. If you happen to have any cuts on your hand, these are a portal of access for microbes. Therefore, any small cut should be covered with an elastic, a plaster. If you have long hair, you must have it tied back behind your back. It's very important that you have a well-organised bench space. You'll be working with both liquid cultures of bacteria and solid cultures. Liquid cultures should be in a test tube rack. Your solid cultures will be in the plates. Organise yourself so that you never have to stretch over a Bunsen to reach anything. In addition, you'll be needing to take notes during the practical. Leave a space to write notes, and remember, you never ever put a plate on top of a lab book. If your lab book's contaminated, we autoclave it to destroy it. Remember, microbes are invisible, and you don't want to risk any contamination. Jane is now going to show you how to work with and handle liquid cultures and solid cultures, containing the cultures in the tube, protecting yourself, protecting those around you, and protecting the sample from contamination.
The labs will be laid out with all the equipment and materials you need for the exercise you're going to do. For example, here you can see we've got an automatic pipetta and sterile tips, some liquid broth cultures, some empty broth tubes, some plates, and a plate culture. Before lighting your Bunsen burner, it's important to check that the rubber tubing attached is securely connected to the gas tap. When you're using it, you can move the Bunsen burner around to make sure it's in an appropriate position for your work, but try not to move it too far and loosen the connection on the gas tap. When doing your practical, it's very important to work in the correct manner. The use of aseptic technique not only protects you and your fellow workers, but also your work. So the first thing we're going to do is light the Bunsen burner, which provides you an area of sterile working space. So move your items if you need to clear a space around your Bunsen. Turn on the gas, close the air hole and light it with the lighter provided. When you're not using your Bunsen, close the air hole and leave it on a nice yellow flame. When it's in use, we open the air hole and have a nice blue cone. It's important to work safely when you're working in a lab. Keep your lab books and your notebooks away from your work, and remember never to place your plates on top of these because there could be a risk of contamination. So try and work as tidily as possible. If you have equipment you don't need till later on in the session, perhaps move it to the back of the bench to give yourself a bit more room. So we're going to show you now how to streak a plate using good aseptic technique. One of the most important aspects of aseptic technique is flaming your loop. You need to make sure that your loop is sterile before you use it, and we do this in a specific way. We put the loop into the top of the flame, and draw it up slowly through the flame so that the loop is the last part to be heated. We remove it from the flame and just allow it to air cool. It's important to remember not to put this down on the bench now, otherwise you'll have to start again as you will have contaminated it. We're now ready to work with our bacteria. When you see an agar plate in the lab, it'll always be presented to you lid down. That enables you to pick it up and have the bacterial side in your hand, ready to work with. When the plates are incubated, they're always incubated lid side down to avoid condensation appearing on the surface of the agar. Take a well-isolated colony from your plate with a cool, sterile loop. Place the agar plate onto its lid. Take a fresh plate and streak as we've shown in the other video clip. At this point it's important to remember when flaming your loop between inoculations that you do not cause any residue that might be present on the loop to splutter. So it's important to draw the loop very slowly up through the flame, especially when you've finished your work. I'm now going to show you how to inoculate from a broth culture. Take your pre-grown broth culture and having a flamed, cooled loop in your right hand, place the lid of the test tube in your small finger. Remove the lid of the tube and flame the neck of the test tube. Place your loop into the broth culture and collect a loopful of culture. Flame the neck of the tube and replace the lid. Always make sure you put this back in a rack to avoid any spillages. We're now going to take a fresh tube and inoculate it. So as before, we remove the lid of the tube with our pinky finger, flame the neck of the tube, place the loopful of culture in the broth, remove our loop, flame the neck of the tube, return the lid and place in our rack. We've now got a loop full of liquid in our loop, which we need to make sure we sterilise without causing any splutter. So we're going to draw this very slowly up through the flame and make sure we don't spit and splutter across the bench. When you've finished inoculating your plates or tubes, it's important to remember that you label them. Label in small writing around the edge of the plate so that you can actually see what's growing on the plate. Label with your initials, the type of media if appropriate, and the date. You should always write on the agar side of the plate. Then put this plate ready for incubation in a basket with the appropriate conditions. Also remember to label any tubes, write on the side of the tubes if possible and not on the cap, and put this in the appropriate test tube rack for incubation. it's extremely important to discard of any contaminated materials that you might have used during your practical in the correct manner. For example, here we have a Gilson tip, which we've used for transferring liquid. We must make sure we place this in the tip discard pot provided. If you have slides that you've used for gram staining, for example, they should go in the pots of pink chloros solution that will be on your bench. Also, filter papers that you may have used for biochemical tests can also be discarded into the chloros. We also use these cotton swabs in some practicals. Those two go into the chloros solution. If you're using a Pasteur pipette, it's also important to put this in the chloros solution, but remove the teat when you're over the pot to avoid any residues spluttering out of the end. Remember, when you've finished working with your Bunsen burner, to turn it off at the gas. Make sure you place all your labelled tubes and plates in the correct basket or test tube rack for incubation. Check the temperature and the conditions written on the label. At the end of your practical, you might have equipment or materials that you haven't used. Ask the demonstrator in your practical what you should do with these items. If you have a spillage of contaminated material in the lab, it's very important that you let someone know so that it can be cleared up in the correct manner. We can see here that someone has laid their test tube flat and the liquid has leaked out onto the bench. So make sure we remove the test tube into the discard tin, and then we're going to deal with the spill left behind. We take this solution of Virusol, which is in a trigger spray, and we're going to spray it onto the area. After 5 minutes, just wipe the area down with some paper towel. We're now going to discard all our unwanted items. You can see here we have a tin that has plastic Petri dishes and agar plates inside it, and we have a tin with baskets that has small bottles and test tubes in it. It's important to remember not to mix these items up, as when they're disposed of from the autoclave, it can cause a nasty mess if the plastic melts all over the glassware. So place your unopened plates into the tin and you're ready to go. Any test tubes should be placed in the tin with the basket. When you've finished working, it's important to swab your bench to remove any trace of contamination that you might have left behind. So we use Tegadyne for this purpose. So we tip a drop of Tegadyne onto the bench, replace the lid, and then using paper towel roll, we swab this over the area in which you've worked. Lift your Bunsen if necessary, and leave the surface damp. The last thing to do at the end of your practical is hang back up your lab coat and wash your hands. Always remember to remove your lab coat first to avoid recontaminating your hands when you take it off. Hang it back up on the hanger and then you're ready to go.